This is a B6 triple content video from the topic Inheritance, Variation and Evolution, and it's discussing cloning. Okay, so this is biology only, looking at two methods of cloning: embryo transplantation and adult cell cloning, which is how Dolly the sheep was created. So let's start with what we know about cloning already. So we've talked about asexual reproduction. This can occur in plants where they don't produce any flowers, and we talked in a previous video about the spider plant, which you can see here, where it makes a tuber, grows a genetically identical copy of the spider plant at the end of that tuber, and if this was in a forest, it would bed down into the soil, creating a clone of the original species. Really good for quick colonization of an area. If we have these plants at school, we can take that extra plant off the tuber, repot it, and we've got ourselves another spider plant. So clones, because they are genetically identical to the original, and that's asexual reproduction. Our second example is identical twins. Identical twins are formed from one egg and one sperm, and once their embryo starts dividing by mitosis, the embryo splits into two, and we get two genetically identical individuals. We can take cuttings from plants, dip them into rooting powder, which contains auxin, our plant hormone, and we can regrow them into pots. And because the cutting is taken from the original plant, it is genetically identical. It is a clone. A nice cheap way of getting plants if we're prepared to wait for them to grow without going to the shop and buying more. Next, it leads us on to embryo transplantation. This is a bit like using a combination of selective breeding and IVF. Okay, so selective breeding, we've chosen this bull and this cow because we think that they've got the characteristics we want, the traits we're looking for, and we will get a really good offspring from them. So that's the selective breeding part of this process. Now, because we don't just want to have one offspring, we want to have several offspring at once, because we're a farmer, we're trying to make good profit, we can use IVF to create these embryos and have more than one. Okay, and the whole process is called embryo transplantation. Let's look at the steps. So a farmer has chosen the individuals that he thinks has the right characteristics to make good offspring. The sperm is taken from the best bull, in this case, and used to fertilise an egg from the best cow. So this is the IVF part. We've selectively chosen them, so that's the selective breeding part, and now we're using IVF to fertilise them together in a petri dish. We'll be watching that fertilised egg divide, form into an embryo, and then the embryo can be separated a bit like it would occur naturally in identical twins, but it can separate into separate embryos, and in this case we've gone for 4 separate embryos. Those embryos are implanted into the uterus, and it's important to use the word uterus, of 4 surrogate cows. Surrogates are basically cows which are going to act as the mother even if they're not genetically involved, so they're going to provide the uterus for the embryo to grow before it's ready to be born. Okay, and the cow then gives birth to the calves, and they are all clones of each other. So rather than having just one offspring and using selective breeding, we've got 4 offspring, a lot more profit to be made, and they're all identical. Okay, problems with cloning is that if they're identical, they can all be susceptible to the same diseases, and eventually, if you keep doing these processes, you can have issues with inbreeding. Okay, so embryo transplantation, selecting the individuals we think have the best traits, using IVF, separating that embryo out and putting it into the uterus of the surrogates. Okay, let's have a look at adult cloning. Okay, this is how Dolly the sheep was created. So in this case, there is no sperm involved at all. Okay, the sheep at the top there, we take a skin cell because a skin cell hasn't differentiated very much. Okay, it's quite a simple, basic cell with the pairs of genetic information. That's the important bit. We need pairs of genetic information, so all the genetic information that we're copying comes from that original sheep that we're trying to clone. Next, we take an egg cell from an adult female sheep, okay? We need an egg cell because egg cells can turn into embryos, but we don't want the genetic information from that egg cell. So we remove the nucleus, and then to fuse the skin cell into the empty egg cell, we use an electric shock, okay? The fertilized egg starts dividing into an embryo, and when that embryo looks like it's going to be viable, it's going to be successful at continuing to grow by mitosis, it's placed into the uterus of the surrogate mother. When that offspring is born, the lamb that's born is genetically identical to the original lamb, the original sheep that the skin cell came from, okay? Because all of the genetic information came from that original sheep, okay? Where the egg cell came from, we took the nucleus out. It was just the empty shell of the egg that we were using to turn it into an embryo, okay? No sperm involvement at all. Hope you found that useful. Thank you.